All right, today we're going to be looking at genetics. So, this is your basic introduction to genetics and what all it entails. So, what is genetics? Well, genetics is the scientific study of heredity, of how traits are passed down to offspring. We're going to look at Gregor Mendel. He was born a peasant in Austria in 1822, and his father was a farmer, and he taught him some of the techniques for artificial selection like animal breeding and plant grafting. And he left the farm and actually became a monk at a monastery in 1843 after attending college and planning to become a teacher. But it ended up he didn't like that job. Even though he taught biology, he had plenty of interest in statistics, he was very good at math. And during his time before he became a monk at a monastery, he also studied at the University of Vienna. But he did return to the monastery where he had spent some time studying plants in his efforts to learn about biology. He returned to this location and he did continue to teach, but he primarily worked in their garden. And he did so between 1856 and 1863, during which he studied heredity and the variation in the common garden pea plant. He actually grew and tested over 28,000 pea plants. And here is an image of Gregor Mendel's garden. So he is considered the father of modern genetics. And that is because even though during his lifetime we did not realize the significance of his work, he is responsible for providing a lot of the data and evidence for how traits are passed down, especially if we consider complete inheritance, which is known as Mendelian genetics. So he studied his pea plants for 8 years, he bred and crossbred them, and he eventually did publish that information, his data, with four conclusions. But again, his work went completely unnoticed until the 1930s, and that was after he had died, which was in 1884. So he never witnessed the significance of his experiments. It was never during his lifetime that he was considered the father of modern genetics. He had no idea that we'd be talking about him in an intro biology class today. So why did Gregor Mendel choose peas? Well, they had easily identifiable traits. Some of the peas were yellow, some were green, some of the plants were short, some were tall. The location of flowers, or wrinkled versus smooth seeds. There were plenty of physical traits that were very easy to discern against. For example, you didn't mix up green versus yellow peas. They were obviously different traits. The pea plants were very easy to grow as well. They're easy to grow and cultivate and crossbreed. And you could work with pretty large numbers of samples, and therefore you could identify and observe great variation during one particular generation. And they also produce large numbers of offspring in a pretty short period of time for a plant, and he could control pollination of the pea plant that he bred and crossbred. That goes back to them being very easy to grow and cultivate. So here are the definable characteristics that Gregor Mendel studied. Some pea plants were tall, others were short. Some pea plants had inflated pods, others had constricted pods. Some pea plants had green pods, others had yellow pods. Some pea plants had terminal pods with flowers at the top, while others had axial pods with flowers along the stem. Some pea plants had white flowers, others had purple or what you might consider violet flowers. Some pea plants had yellow peas, others had green peas. And some pea plants had round peas, while others had wrinkle peas. So you can see his definable characteristics in this chart here on the right. And he actually was able to determine that these readily identifiable traits were being passed down as dominant and recessive traits. So more on that in a minute. So first let's look at Mendel's experiments. Well, he noticed that some plants bred to produce the same characteristic. For example, some tall plants would always give rise to offspring that would end up being tall plants. And when that happened, when over and over again that plant always produced a similar trait, he would say that that plant is said to have bred true. But not all plants bred true. Some tall plants gave us short plants, meaning we had this trait that kept kind of popping back up in the offspring that seemed to be masked or hidden in certain generations. And Mendel knew that there was a tendency for some plants to breed true, while others did not breed true. and that was the key to understanding the mysteries of reproduction and heredity. He wanted to figure out why is one trait disappearing and then showing back up later. What did that have to do with anything? So the first thing Mendel did was create a pure or true breeding generation for every one of the physical traits we discussed. Again, a true breeding organism has a certain characteristic that is always passed down onto its offspring. That's why we can say that this organism bred true with respect to that one characteristic. So he wasn't necessarily creating plants that bred true for multiple characteristics, but he would create a plant that always passed down, for example, violet flowers. That particular trait would show up again and again in the offspring of a particular plant when it was bred. And he made sure that certain pea plants were only able to self pollinate. And that made sure that no stray traits were ever introduced into that lineage. And that made sure that he got really true breeding plants, meaning that if they were only self pollinating, they weren't able to get any other alleles or genes from any other species of plant that had maybe slightly different traits. And that self pollination really made sure that his plants were 100% true breeding. And he did this by cutting away the stamen or the male part of each flower, and then he would control fertilization by using a paintbrush. So here is Mendel's method. This is how he would ensure self-pollination. We can see at the top that we have the stigma, that's the female part of the flower. It would contain near the base of it the ovary. The stigma is just the sticky part that the pollen would stick to, and then it would have to travel down a pollen tube to the ovary. We can also see the anther and the filament. The filament supports the anther, and the anther contains pollen. Pollen contains spermatozoa, or the sperm for a plant. So we would need to get the pollen on the stigma, and it would need to travel down to the ovary. Well, a plant can self-pollinate itself, but that pollen can also be dispersed by the wind or by bugs and move to other plants. To make sure that self-pollination happened, Gregor Mendel would take pollen from a true breeding plant and cross-pollinate it on another plant. Then after that plant produced offspring, he would remove the anthers of the offspring plants and then only use that pollen on that plant with the removed anthers. And that made sure that it was always that plant's pollen that was fertilizing itself. So you can do a couple of different types of pollen transfer, but this was his method for creating true breeding plants. So at this point in time, Mendel didn't know anything about DNA. Nothing about it being a double helix or how traits were actually genes that were being passed down after a process called fertilization. So he just really called his characteristics, but we would now call them traits. And he studied all 7 of his definable characteristics or traits in pea plants. And he was very lucky. He didn't know it at the time, but he was studying traits that were each determined by a single gene. Sometimes a particular trait is determined by multiple genes, or sometimes there's codominance or incomplete dominance, things that we're going to look at. But Mendel got very lucky, and it just so happened that all of his traits were not only determined by a single gene, they all had the same type of genetic inheritance called complete dominance. And Mendel first had ensured that his true breeding generation was pure. His next step then was to cross-pollinate the plants that showed contrasting traits. So he's going to cross-pollinate or breed 2 plants with different traits together and see what happens. The offspring that resulted from this cross-pollination is called the first filial generation, or F1. So here we can see that Gregor Mendel cross-pollinated 2 plants that had different traits. And this process is very similar to creating a true breeding population, but instead of ensuring similar pea plants were produced, we have cross-pollination of different plants showing opposite traits. So for example, he would take a purple flower plant and a white flower plant, and he would cross-pollinate them. He would then take the peas that they produced, plant them, and observe his definable characteristics in that first filial generation. He noticed that when he crossed purple flowers with white flower plants, that he got offspring that all had purple flowers. So it seems that the white trait disappeared. The white flower trait completely disappeared, at least in that first filial generation. And all of these crosses are considered monohybrid crosses. because you're looking at just one trait to do this cross. And another example of a monohybrid cross, like the one we just looked at, is when you cross pure yellow peas, or plants that produce pure yellow peas, with plants that produce pure green peas. He noticed, very similarly with the purple and white flowers, that all offspring were of one type, they were all yellow. But he would still consider these offspring in the first filial generation hybrids because they're the offspring of crosses between parents with contrasting traits. And in crossing the true breeding plants with each other to generate a first filial generation, Mendel discovered that some traits showed up over the other every time. And again, that's how he determined which traits were dominant or recessive, by crossbreeding, for example, purple versus white flowers. The purple flowers always showed up in that first filial generation, so he determined that trait was dominant over white. Tall plants were dominant over short, yellow seeds over green, round seeds over wrinkled, and green pea pods over yellow pea pods, inflated over constricted pea pods, and flower position was dominant at the axial or end of stems that go all the way up the plant instead of just at the very tips or terminal. So you can see here a chart that shows you the results of his monohybrid cross, which were the crossbreeding between two plants that were pure breeding for the same trait, but then he took the two of differing traits, but same definable characteristic like flower color or plant height, and crossed them together. This helped him determine which traits were dominant and which were recessive. So when Mendel allowed then the F1 generation to self-pollinate, the missing traits showed back up. So for example, with the purple and white flowers, we crossed them together, we got an F1 generation, and they were all purple. All the flowers were purple, so we had 100% of the plants having purple flowers. That's what happened in the first generation or first filial generation, F1. Well, then he's going to take one of those offspring, one of the F1 generation offspring with purple flowers, and he was going to self-pollinate it. So he was just going to have it pollinate itself. Well, he noticed that the hidden trait, in this case white flowers, popped back up in the second generation or second filial generation, also called F2. And it popped back up in a certain proportion. It was always 75% the dominant trait with 25% the recessive trait, or at that time it was called the hidden trait or missing trait. So there was a 3-to-1 representation in F2 generation. So thus, if the purple flowered plants produced in the first experiment, F1, were self-pollinated or crossed with each other, that's another possibility, 75% of the offspring of the F2 or second filial generation produced had purple flowers, and then only 25% had white flowers. So what you can see is that in a second generation, a hidden trait can reemerge, but it generally reemerges in a lesser percentile or proportion than the dominant trait. And this may make you think, oh, well, dominant traits are always more common in a population. That's not the case. We really have to look for the specific example of inheritance. In this example, the dominant trait is more prevalent, and we see that a lot in plants. But that does not mean the dominant trait is always more prevalent in a population. And looking again at Mendel's second cross, he allowed the F1 generation to self-pollinate, thus producing the F2 generation, and he saw that the recessive trait showed back up. It was previously missing, so it hadn't completely disappeared. So what happened when he crossed two yellow pea hybrid plants? Well, he saw again in the F2 generation that missing trait reappeared. And so again, 3/4 of the peas were yellow and 1/4 of the peas were green. That's our 75-to-25 or 3-to-1 ratio, looking at the traits or the phenotype. So during the formation of the sex cells or gametes, the alleles must have separated or segregated to different gametes. And that would have produced pollen in an egg that contained different alleles. And when self-pollinated or crossed with each other, the missing trait had a chance to reappear because of the resorting of different types of traits or alleles within a gene. Alright, so here is the 3-to-1 ratio in the F2 generation through all 7 definable characteristics that Gregor Mendel studied. Remember, purple flowers are dominant over white, axial over terminal, yellow seed color over green, round over wrinkled seed shape, pod shape inflated is dominant, pod color green is dominant, and tall plants are dominant over dwarf plants. So he had true bred all of these generations, then he crossed them with similar characteristics but opposite physical determination. For example, purple to white, axial to terminal, yellow to green, round to wrinkled, inflated to constricted, green to yellow, tall to dwarf. These, what we're seeing here in the middle, are his crosses. They're not pure breeding crosses. It's true crossbreeding, where he took two traits that were opposite each other, crossed them together, and in the F1 generation, we saw only the dominant trait. Well, in the F2 generation, if we counted the offspring, which Gregor Mendel did, remember he tested numerous pea plants, and he did this numerous times, he got every time essentially a 3-to-1 ratio. So with statistics, this would have been averaged, but essentially a 3-to-1 ratio every time in the F2 generation. In the F1 generation, the dominant trait showed up, but in the F2 generation, the recessive trait, the hidden missing trait, does reappear. So this let Gregor Mendel conclude that inheritance is determined by packets of information. At the time, he didn't know these packets of information were called alleles, that they were on genes, which are segments of DNA. He called them factors. And he said these factors are passed on from one generation to another. Now again, he had no idea these factors were genes, or that there were different types of genes that could be inherited, and different types of genes were called alleles. He knew nothing about chromosomes or DNA. And why? Well, these terms hadn't been yet defined. They had not been defined. We actually didn't determine until 1953 that DNA was even what was being passed down to offspring. So again, Gregor Mendel was way before his time, and that's why we didn't realize the significance of his data before his death. So he also concluded that each organism has not one, but two factors that determine its trait. And this really underlies a lot of modern genetics. And he called these two factors that determine its trait, basically different types of genes, he called them alleles. And one of a pair of genes that occupies the same position on homologous chromosome, that's an allele. It's a type of a trait. For example, the dominant versus recessive trait, those are alleles. You can inherit one or the other from your parents. Dominant traits, or alleles, was an allele that will determine a phenotype if just one is present in the genotype, meaning if you inherit a dominant trait, it shows up in your physical characteristics. Your genotype is the trait you inherit. The phenotype is the actual physical characteristic or trait it determines in you that others can see. A recessive trait or allele is an allele that will not determine the phenotype unless the genotype is homozygous in that allele, meaning unless you inherit two recessive traits, then you're not going to show the recessive trait physically. You would have to inherit two genes of the same type, both of them be recessive, in order to show that trait. Otherwise, the dominant trait is going to overpower that recessive trait. That idea is called complete dominance. And in sexual reproduction, each parent contributes only one of its factors, or alleles, to the offspring. And if a dominant factor for a definable trait exists in an organism, the trait determined by that dominant factor will always be expressed if it exists in the offspring. So we know now that Mendel's factors are genes, and we know now that genes are passed from one generation to another. We don't have to call them factors. We know there's different types of genes, and those are alleles. And Mendel's second principle came about when he noticed that some forms of genes, or alleles, to him factors, some forms of genes were dominant and some were recessive. He recognized this idea and he said, or wrote down when he was writing his postulates, that this proved what was happening in the F1 or first filial generation and why one of the traits was missing and then could show back up because of this behavior, because of the idea of dominant and recessive traits. So Gregor Mendel was able to recognize that there are alternate factors for each trait. So different ones for flower height, seed color, seed shape, flower color, and he assumed that if he had a true breeding plant, it always breeds true, and pretty much the offspring are always identical. meaning a tall pea plant would always produce tall offspring. In that way, he assumed that his true breeding plant that was breeding true must have identical factors. And that's why when he performed the F1 cross, Mendel was covering up or getting rid of that recessive trait for a generation because this recessive or absent factor was being lost in the dominant true breeding population. But it was essentially not being completely lost. It was only hidden, it was being masked by the dominant or visible trait. This idea led to him coming up with his theory of complete dominance. He didn't know about pairs of homologous chromosomes, and that if you look at the chromosome from the male parent and the chromosome from the female parent, or from the two parents that produce offspring, if we're talking about plants and not humans, that you can see different locations of genes, and that genes were just segments of DNA. And sometimes those genes matched, sometimes they didn't, and we've now developed terminology to talk about what it means. If the genes are at the same location, they're the same shape, and they match, they're the same genetic information to be inherited. For example, if both chromosomes carry the same allele of the gene at a locus, which is a location on a gene, or specifically a chromosome, then the organism is homozygous at this locus. If the locus contains another gene for which the organism is homozygous, then you'll see at least two traits that are the same size and shape. In this case, in this illustration, color as well. But if each chromosome carries a different allele of this gene, so the organism is heterozygous at this locus, then you'll see different traits at a location. So the term homozygous means to have the same allele or same type of gene on a pair of homologous chromosomes you've inherited from your parents. The term heterozygous means to have inherited two different versions of a trait from your parents. And we can predict genetic probability crosses. We can do so by doing something like a Punnett square, which is basically a sophisticated coin flip. We're basically figuring out the likelihood of a particular event occurring, and that does really fall down to chance, but it can be expressed as a fraction or a percent. So coin flip again, it's a great example. Now, Reginald Punnell developed the Punnett square. It's a basic diagram used to show the probability or chances of a certain trait being passed from one generation to another. And gametes, or in this case we're going to use letters, they are always placed above and to the left of the square. And then in the squares, that's where we're going to put the letters of the offspring. So the gametes are going to be the letters you're inheriting from your parents, and the ones in the boxes are going to be the letters that we could possibly have a combination of and produce offspring. We're going to use letters as the alleles, and for the dominant allele, we're going to use capital letters. For the recessive allele, we're going to use lowercase letters. So in this case, we're using Y. So capital Y would be dominant for a particular trait. Lowercase y would be recessive for a particular trait. All right, so here let's look at how we would generate the gametes. So we have at A, we have a parent that can only donate the dominant trait, because that's all they got from their parents. They only have capital A and capital A, or the dominant trait at this location for a particular trait they could pass down. So the gametes they could produce are capital A or capital A from their two different homologous chromosomes. Whereas if we look at B, this parent is heterozygous or has two different traits for this particular factor that they're passing down. So they could pass down a capital A or a lowercase a. So we have to consider everything the parents could pass down in our Punnett square. So here is an example. So we have one parent that had capital Y, or the dominant trait on one of their homologous chromosomes, and a lowercase y, or the recessive trait, on the other homologous chromosome. And the other parent was the same. So we have capital Y, lowercase y for both parents. That's what they could donate. So capital Y, lowercase y, we put one of the parents up top. Capital Y, lowercase y, we put the other parent on the side. And then we bring everything up top down, everything on the side across. So here in this box, we have capital Y, capital Y. Here in this upper right-hand box, we have capital Y, lowercase y. Now, if you're getting used to Punnett squares, you could write lowercase y, capital Y. It's just general convention to always write the capital first if it's there. Here we have capital Y, lowercase y. Here we have lowercase y, lowercase y. So what we're seeing here is that with two heterozygous parents, so they have two different types of traits for the same physical factor being inherited, that produces a 3-to-1 chance for the organism to be either yellow or green. So here we're looking at seed color. That makes sense because seed color, specifically yellow seed color, is dominant. So if you had a capital Y, you show the dominant trait. If you have a capital Y, lowercase y, since you have that capital or dominant trait in your genes, you still show the dominant trait. So only if you have two of the recessive trait, or you're homozygous recessive, would you show the green trait. So again, homozygous is when an organism possesses two identical alleles. You could be homozygous for the dominant trait or homozygous for the recessive trait. If you're heterozygous, that's when an organism possesses different alleles. And so heterozygous, there's not two different types. It's just the heterozygote. We've also briefly talked about genotype and phenotype. So genotype is the letter set we're using. The actual letters that represent alleles of an organism, whether dominant or recessive, is the genetic makeup. And again, we're using letters to visualize the genotype. The phenotype is the actual trait it would give you, the observable expression of an organism's genes. And this is a physical appearance. So genotype is the genes, phenotype is the physical appearance, the expression of the trait, like yellow versus green seeds, or short or tall, or purple versus yellow flowers. And speaking of probability and statistics, no one event has a greater chance of occurring than another. You cannot predict the precise outcome of an individual event. The more trials performed, the closer the actual results to the expected outcome. And what that means is if we go back to this Punnett square example, just because there's a 3-to-1 chance of offspring having yellow versus green seeds, does not mean that, for example, if the offspring produce green seeds, that then the next time they're crossed, they have to produce yellow because that's all that's left. Every single time these parents produce offspring, they could produce offspring with the yellow trait or the green trait. They could produce offspring that have dominant traits only, or are homozygous dominant. They could produce offspring that have the heterozygote genes, or they could produce offspring that are homozygous recessive. So just because parents have offspring of a certain type doesn't mean the next time you do the Punnett square, you remove that option. That option is always available every time they have offspring. So here's another Punnett square example. So here you see on the left, the mom and dad both can only donate the capital letter B. So we could even shorten the Punnett square to one block because the only option for their offspring would be capital letter B. Same can be said for the example on the right. They can only donate these parents small letter b. So their offspring could only be small letter b. Here's another example with the heterozygous parents again. So this mom could donate capital B or lowercase b. This dad could donate capital B or lowercase b. So we have to actually spread out the genes on the top and side of the Punnett square, bring the top down, the sides across, and that would give us, for example, phenotypically, a 3-to-1 ratio. So we'll say that these kind of shaded in eyes are sunglasses. So 3 of the offspring would or could have sunglasses, and 1 wouldn't. 2 of the offspring would be heterozygotes, 1 would be homozygous dominant, and 1 would be homozygous recessive. So for example, looking at this, if I asked for the phenotypic ratio, you want to give me the ratio based on traits. Well, looking at the traits, it's 3 sunglasses to 1 without sunglasses. But if I ask for the genotypic ratio, we want to look at the genes, the letters. So it's a 1 possibility for homozygous dominant, a 2 for the heterozygotes, and 1 for the homozygous recessive. So you could say that the genotypic ratio was a 1 to 2 to 1. You're looking at all the options genotypically. So again, a restatement of Mendel's principles is what we need to go through next. So we want to combine information not only that Gregor Mendel had, but that we now know about genes and traits and homologous chromosomes, and that is that the traits of an organism are determined by its genes. Each gene has an alternative form called an allele, and each organism has 2 alleles that make up the genotype for a given trait. Now, those 2 alleles can be the same, or they can be different. If the alleles are the same, the organism is homozygous for that trait. If the 2 alleles are different, the organism is heterozygous for that trait. And in sexual reproduction, the gametes that could combine do so in a process known as fertilization. We do know that the separation of alleles in parents that occurs during meiosis in order to form the gametes that will come together in sexual reproduction, that idea of that separation of the alleles is known as Mendel's law of segregation. The idea that the union of gametes during fertilization reunites allele pairs and offspring randomly. And if a dominant allele for a genotype exists in an organism, the phenotype is determined by that allele. And this is known as Mendel's law of dominance. So Mendel recognized the idea that during meiosis, gametes have their alleles split. So that's the law of segregation. His law of dominance says that if one of those alleles that an offspring inherits is dominant, then they will show that dominant trait physically. Their phenotype will represent that dominant trait because the dominant trait masks the recessive trait. That's Mendel's law of dominance. We also want to talk about independent assortment. So his crosses demonstrated that chromosomes do truly sort independently of each other during meiosis. So during meiosis, in order to create the 4 gametes that result, we have an independent assortment or a random separation after we split the sister chromatids. He was able to show this by doing a dihybrid cross, or looking at 2 traits at once. So again, this is best seen, the independent assortment is best seen in the 2-factor cross called a dihybrid cross. He specifically looked at color and shape of peas together, and he had an F1 cross to produce the F2 generation, and he used one parent that was homozygous dominant for both traits, one that was homozygous recessive, and this resulted in offspring that had round yellow-seeded plants and wrinkled green-seeded plants as parents. And the offspring ended up being all hybrids, again, in the F1 and even F2 generation, that were heterozygous for both traits, meaning the dominant trait was shown, but genotypically, they did have the recessive trait in their genes. Now, to figure out independent assortment, you can look at this basic chart. Meaning, if we're looking at 2 traits, we have to take the 2 traits that are being looked at. In this case, A, capital and lowercase, B, capital and lowercase is going to be the genotype of this dihybrid. Meaning, we're looking at 2 traits in one organism that are both heterozygous. And you just want to see all the possible combinations that could occur. Capital A could go with capital B. Lowercase a could go with capital B. Capital A could go with lowercase b. And capital A, or lowercase a, could go with lowercase b. And that produces 4 possible gametes. And this was the best way that Gregor Mendel found to demonstrate independent assortment. The idea that gametes do separate separately. And here we can see another dihybrid cross. I'm not going to ask you guys to do any dihybrid crosses, but I want you to know that a dihybrid cross looks at 2 traits at once. So we'll have to look at all 4 gamete possibilities from that combination. And looking at it here, if we want to look at the phenotype, the actual physical trait, if both traits are dihybrid and heterozygous, you always get a 9:3 to 3:1 phenotypic ratio. If the parents are both heterozygous for both traits, the phenotypic ratio is always 9:3 to 3:1. So again, a monohybrid cross is a cross between 2 individuals concentrating on only one definable trait. A dihybrid cross is a cross between two individuals concentrating on two definable traits. And Mendel published his paper on heredity in 1866. Again, that was before we knew a lot about genetics or even DNA and genes and alleles, and the scientific community saw little, if any, importance in his work. He died in 1884 with no recognition for his contributions to genetics, but today we do know him as the father of modern genetics. Because since Mendel's time, we found out that some alleles are neither dominant nor recessive, that many traits are controlled by more than one gene. That's called polygenic traits. And that we know now that there are more complex inheritance patterns. There's not just complete dominance, which is what Gregor Mendel discovered and studied. Complete dominance is that for each of the character traits that Mendel studied, there were only two alleles, and each exhibited a dominant or recessive trait that would be passed on to offspring. This idea of dominance always masking the recessive trait, that's complete dominance. And Mendel again got lucky that all his traits were inherited the same way, so that both the homozygous dominant individual and the heterozygote always express the dominant phenotype. Because if the dominant letter shows up in the genotype, that's the phenotype that is shown. But we now know that is not always the case. There are more complex inheritance patterns: incomplete dominance, codominance, multiple alleles, and polygenic traits. And we're going to talk about these next time.